about male density and rapid evolution of genital morphology in a sea beetle. High male competition, as we know, increases selection on male reproductive traits, and this can be true from behavior to the organ level to the cellular level. We also know that, particularly within insects, male genital morphology is extremely diverse, and in fact, some biologists who are taxonomists will classify insects based on their genital morphology alone, particularly when other somatic traits are nearly identical. So I wondered, can intense male competition influence the evolution of male genital morphology over several generations? And I utilized Colossobucus maculatus, which is a bean beetle, pictured here. This is actually a male picture. The males and females are anatomically dimorphic, where you can easily distinguish males from females. So here is a male. They tend to be smaller and shorter in length than females, which are larger. The females, they have internal fertilization, and then the females will oviposit eggs on top of beans. This is actually the type of bean that I use. It's called a mung bean. The larvae will develop, burrow into the bean, and develop into adults. This process can take as little as 3 weeks, so generation time is about 3 weeks. And then they emerge as adults, and they live for about 2 weeks, and they don't require food or water, so they're extremely easy to use in the lab. I created 2 treatments. I had a monogamous treatment where there was 1 male and 1 female, so forced monogamy, and a polyandrous treatment where I had 6 males and 1 female, so extreme, intense male competition, and I did this for 7 generations. Now the male beetle genitalia looks like this. The testes are here and internal, so this has been extracted from the beetle. Then the genital intromittent organ, which is external, has these spikes on the end. So we had an SEM, so we did electron microscopy on the very end of this intromittent organ and measured spike length over the course of several generations. Now this particular data that I'm going to show you was part of a much larger data set, and in fact was sort of a byproduct. So unfortunately I don't have data from each and every generation, but I do have snapshots. So in generation 3, the light gray bar is the polyandry treatment, and the dark gray is monandry. So in generation 3, we already saw an increase in the spine length, and in fact increased to generation 7. And there's a reason for this large error bar, which if you see me later, you can ask me about. Okay, so what we saw was that males in the polyandry treatment increased spine length from 50 microns to 63 microns, which ended up being significantly longer than the monogamous spines. So in this particular species, spines will puncture the female reproductive tract, and in fact, generally the reproductive tract is inward on itself, and when they mate, the spikes will come out and damage the female internally. There was actually a previous study that had shown that. And they transmit seminal fluid this way, so the sperm are not transmitted through the spikes, but the spikes actually do transmit seminal fluid into the hemolymph and is thought to reduce remating and alter female physiology. This work was conducted at Beloit College, where I was a visiting assistant professor with the help of my 2 students, Willow and Cheyenne. But if you have more questions about this, I will be starting a tenure track position at the University of Dallas this fall. Thank you.